Okay, all nucleated cells express that, and you will understand that later on why. You have also HLA class 2 or MHC molecule class 2. They are called HLA-DP, DQ, DR, and they are present only on antigen-presenting cells, which are dendritic cells, macrophage, and B cells. So if I ask students to tell me which MSC molecule are present on the liver cells, you're all gonna say MHC class 1, fantastic. If I ask you which molecule, MHC molecule, are expressed on lung epithelial cells, you're going to say MHC class 1. If I say now which MHC molecule are present in macrophage, Debbie, can we have a sensing on that?
Yep. You want a poll?
Yes, please. MHC molecule present on macrophage, for example. Because I ask that because students always do the mistake, and I don't want them to do the mistake at the exam, so I'm preparing them for it.
Oh, sorry. I'll have to think about this. Molecule on macrophage.
MSC molecule are expressed on macrophage, and you say answer would be MSC class 1, MSC class 2, or MSC class 1 and MSC class 2.
We are getting answers coming in, all right, on the chat, but let me put the poll up.
Yeah, absolutely. I really like students being engaged. That's fantastic. That's very good on you.
So the poll is up, and people are responding. Okay, so at the moment we've got 2 with MHC saying MHC class 1.
Yes.
2 and 89 saying MHC class 2, and 180 saying both.
Yes, excellent. So that's the right answer. Why? If I say macrophage, they have a MHC class 2, but because they are also nucleated cells, they will also express MHC class 1. So every time students have this mistake, and I want to correct that, I want to warn you that yes, they do express MHC class 1 and class 2. So don't do the mistake. Thank you very much, Paul. So, and one thing very important, they are this gene encoding for this MHC molecule are inherited in block, which we call haplotype, and therefore you're going to have a maternal haplotype and a maternal haplotype, okay? And you will see the importance of that. Okay, so and this is come back to that. What are the features of the MSC class 1 and 2? They are co-dominantly expressed, meaning that you're going to express both the MSC molecule that you inherited from your father and that you inherited from your mother. So this is what we call co-dominance. And you will see this is extremely why, because by having a variety of the MHC molecule, you can present a variety of microbe, and therefore you can mount a response against a different microbe. And this is to keep you in mind with the love story that is coming down afterward. So they are highly polymorphic, meaning that all the genes encoding for the MHC molecule there's different allele, okay? We estimate that there's around 14,000 different MHC molecules, which is amazing, and none of them are identical from individual. And you will understand the importance. And therefore, because we have this highly polymorphic gene, we'll be able to present the microbe differently. And you will see the impact later on. So, which means that if there's a new microbe or a mutated one, like viruses, this polymorphism ensure that we will not be eradicated, that there will always be some people who will mount an immune response, okay? They will be immune to it. Imagine if you had all the same immune MHC molecule, if there's a microbe, we're either dead or live. And because of that, there will always be individual living. And the best example, if you will go back to the Spanish flu, out of an estimated of 500 infected by the virus, 50 million died. 500 million, 50 million died. Not all died, only 50 million, only 10%. So the rest had the MHC molecule that allowed them to mount a decent immune response that were able to control the virus. So this is how you see. And what they do, this MHC molecule, and this is where you need to know, the MHC class 1 on the antigen-presenting cells will present the intracellular microbe peptide. The MHC class 2 will present the extracellular microbe. I'm going to summarize that here. So these are all the viruses we've seen, or you're going to see. What they do is when they've been captured by the antigen-presenting cells, what they do, they're going to use a pathway called endogenous pathway, which is a way to process the virus, the viral protein, and present it to MHC class 1. Forget about this now, the MHC class 2, because it's a bit confusing for later. So just remember, viral protein expressed on MHC class 1 on host cells, but on antigen-presenting cells, this is cross-presentation, and this is the wonder of these antigen-presenting cells. They can process the virus via MHC class 1 and 2. You will understand in my second lecture why I do that. While the bacteria, we've seen this nasty bacteria, they are processed by the antigen-presenting cells by exogenous pathway, which leads to this bacterial peptide to be presented by MHC class 2 only. Okay, so, and you can see that the way the microbe is processed determine the kind of MHC molecule that will use that will be used to present the microbe. So this is how the endogenous pathway, this is how viral protein are being presented by MHC class 1. I'm not going to go too much in details, but just to let you know that viruses get inside the cells. It doesn't need to be a phagocyte, because that's the nature of virus. They infect the cells, and they don't infect any cells. They infect specific cells. But in the case of antigen-presenting, they can infect the antigen-presenting cells as well, because the antigen-presenting have a way to catch them. And once they are inside the antigen-presenting cells, expose this genetic material, start producing viral protein. Because the host knows that this protein are not host, is going to tag them, meaning that they're going to be marked for destruction by this big enzymatic complex called proteasome. And what this proteasome will do is chop the viral protein in very small pieces called antigenic peptide that will be transported to the ER by certain protein. These are transporter associated with processing, antigen processing, TAP. They get into the ER. And in the ER, there's this different MHC class 1, A, B, C that are waiting. And when you have the right match, meaning the right peptide for the right MHC molecule, you have a complex, then this complex is expressed at the cell surface. Okay? Very ingenious system. Okay? So you depend on the right match, and we're going to talk about it. And this right match here will expose will be exposed at the cell surface, and will tell the T cells, look, this is a piece of the microbe that is inside me. This is a virus. This is how it looks like. And the T cells is here have a receptor for that. They will sense the peptide. They will sense the MHC molecule class 1, and the T cells at this point will know exactly what to do. So if this is an antigen-presenting cells, it's going to be activated. If this is a virally infected cell, it's going to kill it. Let me explain that better. So you have a virus, let's say it's coronavirus, gets into the lung epithelial cells. So the first to be infected would be the lung epithelial cells, as you can imagine, and you're going to be cell damage, and ultimately the virus here will get picked up by the antigen-presenting cells. So remember, these viruses here will process these epithelial cells will process the viral virus the same way the antigen-presenting cell does, which is via the MHC class 1. So what you have here is this infected epithelial cells expressing the virus. I mean, peptide, viral peptide, MHC class 1. In the meantime, this antigen-presenting cell, they need to do their job. Why? They need to activate T cells. So what they do, they sense the virus, pick the virus, process it via the exogenous pathway, expresses via the MHC class 1, and go and activate naive T cells that will become infected cells. Where? In the lymphoid tissue. And what these naive T cells become, they become effector cells. They know what to look for because they've been introduced to what the virus look like, and they're going to start looking in the lung epithelial cells which cells have been infected. And the cells have been infected as the one that expressing the MHC class 1. And once he senses that, what it is going to do is going to kill the viral infected. Very ingenious system, no bystanding effect. It's really specific, okay? And the reason why I mentioned that viruses can be picked up by antigen-presenting cells, but also by the favorite target cells, is that every virus has or infect a particular cells, which we define as tropism. Hep B, Hep C infect the liver, lung epithelials, coronavirus infect respiratory epithelial cells, so and so on and so forth. So they have a tropism, meaning that they can infect only this particular cell type or tissue. Okay, and in order for this tissue to get cleared, you need activated T cells. So I hope you understand now the importance of having MHC class 1 on all cells as well as on antigen-presenting cells. So let's see now how antigen-presenting cells process extracellular microbe, meaning bacteria. Look, and everything comes back to where the microbe is processed. Under no circumstances, this is your bacteria, you will see the bacteria inside the cytosol, like you've seen in the case of viruses. They will always remain inside vesicles, and these vesicles will be exposed to lysozyme, which will degrade the microbe. And then this vesicle containing the bacterial peptide will fuse with other vesicles that contain MHC class 2 molecules, which are DP, DQ, DR. And when you have the right match, meaning the right peptide for the right MHC molecule, you're going to have assembly, formation of this complex that will move at the cell surface. And therefore, you have at the cell surface an MHC molecule class 2 expressing a bacterial peptide. And what this will mean that when you have a T cells, in this particular case, a CD4 T cells, you will see that there's a bacterial peptide coming from Staphylococcus aureus, and you know exactly what the CD4 positive T cells will do. And we'll see that in the next lecture, okay? So this is extremely important. So you can see that the location of your microbe determine whether it's being expressed by the MHC class 1, like in viruses being present in the cytosol, or in vesicles being presented in MHC class 2. And you can see that it's an ingenious system to make sure that we develop the right immune response against viruses or against extracellular microbes such as bacteria. So I'm not going to spend too much time on that. These are what they look like, the MHC class 1, MHC class 2. This is basically where the peptide is being presented. I'm not going to say that. And this area where the peptide is presented is the highly polymorphic protein residues. And I said that. So this is a broad specificity, meaning that although one MHC molecule can only present one peptide at a time, one single MHC molecule can present different peptide, okay? And this is just to maximize the presentation capability. And as you can see, this is extremely important. Our survival depends on what? On the ability of MHC molecule to present the peptide. If we fail to do that, then we fail to mount an immune response, and therefore you end up with a very, very serious life-threatening condition. Unless you're being treated, you're most likely going to die. And this is a point that I want to raise, but I'm going to make it more clearer in the next lecture, is that the MHC class 1 only present peptide to CD4 positive T cells, also known as T helper, okay? While MHC class 1 will always present to CD8 positive T cells, which later on you will see will become cytotoxic T cells. These are the T cells that we kill all the cancer cells that we've seen in the patient, or viral infected cells, okay? And just remember the rule of 8. 1 times 8 equals 8, 2 times 4 equals 8. So this is how you remember. If I ask you which MHC molecule present to CD8, you know it's class 1. Which MHC molecule present to CD4, it's 2, because 2 times 4 equals 8. Just a tip to remember. Okay, so these are the point. The host can deal with a variety of different microbes, and this is extremely important. These genetic polymorphisms guarantee that at the population level we are not